This is our project on transgenic bacteria. So basically what is that? It is a bacteria that carries artificially inserted DNA from a host eukaryotic cell. So to start, let's talk about the structure of DNA. There are two main parts, chromosomal DNA and plasmid DNA. The plasmid DNA is separate since it can replicate independently. So basically the desired gene is isolated from a eukaryotic cell, such as a human cell, and that gene is added to the bacteria's plasmid. Then the plasmid is added back into the bacteria's cell to replicate. Think of it like a syringe taking one thing out from another and adding it to something else. Once it is done, it is added into a controlled environment such as a petri dish to grow and replicate. The great thing about bacteria is that for genetic manipulation is that it reproduces rapidly and it's genetically identical at each time. Let's look at this into further detail. So basically the desired gene added to the plasmid needs to know where to insert into. That's where the LAC-Z gene comes in. It basically codes or flags down an enzyme called restriction endonuclease that is used to remove the LAC-Z gene in order to make room for the desired gene from the host cell. Once that is finished, the plasmid is added back into the bacteria with the desired gene for replication. Now let's look at it in real life. Did you know that insulin for diabetics is used in transgenic bacteria? So here's the human and here's the pancreas. Let's zoom in on the pancreas. So insulin is normally produced in the pancreas and then is released into the bloodstream to reduce high glucose levels. But diabetics cannot do this. Since they have an insulin deficiency, they must take regular injections of the hormone insulin. There you can see insulin is being added into the bacteria's plasmid and then is being added back into the human being to reduce glucose level. Another example is the human growth deficiency. Basically, people lack the ability to grow normally because the pituitary gland used to produce growth hormones does not produce enough hormones for normal growth. Therefore, a hormone is given that helps the person with the deficiency grow normally, which is most commonly used in dwarfism. Another really cool example is actually found in transgenic plants, not bacteria. So bananas are actually genetically grown to obtain a vaccine. The banana provides easy means for delivering a vaccine to children since it is edible. Let's look at some controversies with transgenic bacteria. So before 1995, insulin for diabetics was obtained and purified from slaughtered animals, such as cows and pigs. This was a very expensive process and caused many allergic reactions since insulin from animals is slightly different from humans. Today we use insulin from humans. Another one is that the human growth hormone used to be extracted from human cadavers, and that caused a greater risk of transmitting disease such as AIDS and hepatitis C. Now we use transgenic bacteria, which is a lot safer and leads to no transmission of diseases.